Good evening all. So the next part of this microscope is after bright field microscope. Now we are going to discuss about phase contrast microscope. This is a special type of optical microscope whereby small light deviations known as phase shifts, small light deviations known as phase shifts which occur during light penetration onto the unstained specimen. So mainly phase contrast microscope we can view unstained specimen, and this may occur through small light deviations known as phase shifts, and these phase shifts are converted into the image to mean. When the light passes through opaque specimen, the phase shift helps to brighten the specimen, forming an illuminated image in the background. So, the phase contrast microscope, it produces a high contrast images when viewing a transparent specimen more. So, those of the microbial cultures, thin tissue fragments, cell tissues and subcellular particles are viewed using phase contrast microscope which produce high contrast image. The principle behind the working of phase contrast microscope is the use of an optical method to transform a specimen into an amplitude image that viewed by the eyepiece of the microscope. So the principle behind the working of phase contrast microscope is optical method to transform a specimen to amplitude image. The phase contrast microscope we can use to view the unstained cells, also known as phase objects. Unstained cells or phase objects, which means the morphology of the cell is maintained and the cells can be observed in their natural state. So as usually we observe these specimens after heat fixing or drying, then applying stains, etc. Using this method, we can view the cells in their exact morphology and in their natural state in high contrast and efficient clarity. This is because if the specimen are stained and fixed, they kill most cells, a characteristic that is uniquely undone by the bright field light microscope. So this is the characteristic that is uniquely undone by the bright field microscope. The shift that occur during the light penetration, it can be converted to what? The change in their amplitude, and this change in the amplitude which causes the image contrast. So coupled with this contrast enhancing elements such as fluorescence, they produce better visuals for the specimen's image. If we are coupling this phase shift along with fluorescence, we get a more better image of the specimen. Now coming to the parts of the phase contrast microscope. The instrumentation of the phase contrast microscope is based on its light pathways from receiving the source of light to the visualization of the image. Therefore, it is made up of light source, that is mercury arc lamp, so then collective lens, aperture, condenser. These are the parts of the phase contrast microscope. Condenser annula, specimen, objective, phase plate, deflected light and phase ring. So these are the different parts of a phase contrast microscope. So these two are condenser which are placed on the bottom. Then specimen. This is the specimen and these two are the objectives, and these are the which direct the surrounding light, that is the diffracted light and the image plane. The functioning of the phase. Next is the functioning of the phase contrast microscope. The change which are caused by the deflected light or deviated or scattered light and the undeviated light. So there are two types of light which passes both deflected and undeflected light, and when it reaches the specimen, which is absorbed, and this creates a certain wavelength producing color. So the difference created by both light and that of the absorbed variation, and this may create what? Phase shift. അങ്ങനെയാണ് ഈ phase shift. അതായത് രണ്ട് ടൈപ്പ് ലൈറ്റ് പാസ് ചെയ്യും. ഒരു ലൈറ്റ് സ്കാറ്റർ ചെയ്യും, മറ്റത് ഡിഫ്ലക്റ്റ് ചെയ്യാതെ നേരെ പോവും. ഇത് രണ്ടും കൂടി അവരുടെ രണ്ടിൻ്റെയും കൂടെ എന്താ പറയുക, ഡിഫറൻസ് create an amplitude variation. This amplitude variations are allowing the visualization of photographic equipments like phase contrast microscope, which can be seen with our human eye. The condenser of the phase contrast microscope. Now coming to the condenser, it has an opaque disc that is known as annular ring. The most important part is annular ring with a transparent ring, and it possesses a transparent ring, and that transparent ring adiludeyanu it produces a hollow cone of light that passes through this. So it has a transparent ring, and through this ring the light passes. Due to the light variation, some light bend at the specimen. But ചില ലൈറ്റ് സ്പെസിമെനിൽ എത്തുമ്പോൾ ബെൻഡ് ചെയ്യും, caused by the variation in the light density, and it forms an image at the objective lens. Also, there are some light which doesn't bend or undeviated light. They may strike the face ring. അപ്പോൾ അത് നേരിട്ട് ഫേസ് റിങ്ങിൽ പോയി സ്ട്രൈക്ക് ചെയ്യും ഓൺ ദ ഫേസ് പ്ലേറ്റ്. ആൻഡ് ഡീവിയേറ്റഡ് ലൈറ്റ് ആണെങ്കിലോ ഈ ഫേസ് പ്ലേറ്റിലുള്ള ഫേസ് റിങ് മിസ് ചെയ്യും. അപ്പോൾ അൺഡീവിയേറ്റഡ് ലൈറ്റ് ആണ് ഫേസ് പ്ലേറ്റിലുള്ള ഫേസ് റിങ്ങിലൂടെ പാസ് ചെയ്യുന്നത്. ഡീവിയേറ്റഡ് ലൈറ്റ് അതിലൂടെ പാസ് ചെയ്യില്ല. ദേ മിസ് ദാറ്റ് ഫേസ് റിങ് ആൻഡ് ദേ പാസസ് സിംപ്ലി ത്രൂ ദ ഫേസ് പ്ലേറ്റ്. ഫേസ് പ്ലേറ്റിലൂടെ പാസ് ചെയ്യുമെങ്കിലും ഫേസ് റിങ് വഴിയല്ല അൺഡീവിയേറ്റഡ് ലൈറ്റ് പാസ് ചെയ്യുന്നത്. ആൻഡ് ഇറ്റ് ഡയറക്ട്ലി ഇറ്റ് ഫോംസ് എൻ ഇമേജ്. അപ്പോൾ രണ്ട് ടൈപ്പ് ലൈറ്റ് ആയി അല്ലേ, deviated and undeviated. Now, the phase contrast microscope is designed with an objective lens that have the ability to perform multiple functions when combined with contrast enhancing technique. For example, fluorescence. ഫ്ലൂറസൻസും കൂടെ നമ്മൾ ആഡ് ചെയ്യുമ്പോൾ നല്ല കോൺട്രാസ്റ്റുള്ള ഇമേജ് കിട്ടും. The objective lenses are located in the internal face plate with variation in the light absorption and phase displacement, that is undiffraction, creating a wide spectrum for contrasting the specimen and forming a strong contrast in the background. അപ്പോൾ ഈ രണ്ട് ടൈപ്പ് ലൈറ്റ് ഉള്ളത് കൊണ്ട്, അതായത് ഒന്ന് ഡീവിയേറ്റഡും അൺഡീവിയേറ്റഡ് ലൈറ്റ്, അപ്പോൾ ഒന്ന് ഫേസ് റിങ് വഴി പാസ് ചെയ്യും, ഒന്ന് മിസ് ദ ഫേസ് റിങ്. അപ്പം അതുകൊണ്ട് ദിസ് മേ ക്രിയേറ്റ് എ വൈഡ് സ്പെക്ട്രം ഫോർ കോൺട്രാസ്റ്റിംഗ് ദ ഇമേജ് ആൻഡ് ഫോമിംഗ് a strong contrast in the background. Now coming to the applications of phase contrast microscope. It helps in determining morphologies of living cells such as plant and animal cells, and it helps to study the microbial motility and also structures of locomotion. ഈ ലൈവ് സെൽസ് ഫേസ് കോൺട്രാസ്റ്റ് മൈക്രോസ്കോപ്പ് വഴി കാണാൻ സാധിക്കും. അതുകൊണ്ട് തന്നെ this can be used for observing the microbial motility and structures of locomotion. And it also helps to detect certain microbial elements such as bacterial endospores. So in the next class, we can discuss about dark field microscope. So these are all about phase contrast microscope. Thank you all.